In this video, I will give you an overview of how the experiment summary page looks like on the ENCODE portal at encodeproject.org. We organize data in experiment objects. For this tutorial, I have two example experiments to show you today. Let's go to the first one. This is how the experiment page looks like. It has many sections, but we'll break it down together. On the left of the summary page, you will find the summary section. This includes metadata about the experiment itself, for example, the assay, the target, and links to any controls. On the right side, you will find the attribution section. Here, you will find the information on the lab that performs the experiment, links to any corresponding geo entries, and if the experiment was a part of any groupings. If you scroll down, you will find the replicates table. This lists replicates and provides any information on the biosample and antibody used. Below that, you will find the file section. This is separated into three tabs: the genome browser, the association graph, and the file details. On the first tab, you will find the integrated balance browser that is GPU-based and allows you to visualize the tracks. On the second tab, association graph will allow you to track your data provenance, especially how the files were derived from each other. Here in this particular experiment, started from the raw data reads and used to process alignments and peaks. If you click on the blue rectangles, you will get details on this particular analysis step. Clicking on the yellow rectangles will give you more information on the file itself. Clicking on the green circles will give you the quality metric of that file. Also in the association graph, you will notice gray arrows. These will highlight the arrows of the inputs and its outputs. This way you can easily trace the analysis step or where exactly the file was processed during the pipeline run. The last tab, the File Details tab, contains all the raw and processed data in a tabular format. Here you can see the small icons that will allow you to download a specific file of interest within an experiment. In this section, we also have buttons that will allow you to visualize data outside of the portal, such as the UCSC Genome Browser. It is located on the top right-hand corner. Select for the assembly of your interest, the external browser that you would like to see it in, and click Visualize. What all three tabs have in common is the file facet on the left. Here you can pick the assembly of your choice, the file format, or sort and filter by the output type. If you scroll down to the very bottom of the page, you will find all the documents relating to this experiment compiled here. Here I have covered the first example. Let's look at the second example. This looks very similar to the first one, except for the audit icons on the top left. These are used to flag experiments with metadata inconsistencies or if the data is not compliant with standards. These will help wranglers working with the lab to correct the audits. If you click on the audit icon, you will find the reasons for why this particular experiment was flagged. Clicking on the plus icons will give you more information and details within the category. This is all I would like to show you for the experiment summary page. If you'd like to follow this tutorial in an interactive step-by-step walkthrough, I suggest you use the Help widget on the bottom right of your browser window. Look for the Experiment Summary page and Experiment Overview. If you click on that, a series of balloons will guide you through the page step by step.